Hello, everyone. My name is Arielle Schwarzberg, and I'm going to be a rising senior at the Oxbridge Academy, and I spent my summer in the Fitzpatrick lab. And today I will be talking about reconstructing the complete morphology of a developing neuron. So for my project, I studied the morphological development of a neuron to further understand how the functional changes during development could correlate with any structural changes. I compared the morphology of a developing and adult tree-shrew neuron within the visual cortex. We observed the tree-shrew neuron specifically because they have a highly developed visual system similar to primates. And this over here, this is an image of a tree-shrew. So for some background, I want to start with the basic structure of a neuron. Here we can see a cartoon example, which is typically seen in most textbooks. There are also many different types of neurons, such as sensory and motor neurons. For my project, I am working with excitatory sensory neurons within the visual cortex. Over here, we can see the soma, or the cell body of the neuron, and this contains the nucleus. Over here, we can see the axon, and this is what sends signals to other neurons. And then over here, we can see the dendrite, and this typically receives and processes incoming information. Although it's not pictured here, the neurons that I worked with contain two different dendritic domains consisting of apical and basal. On those neurons, there are subcellular structures where synapses occur. The presynaptic side of the neuron is the axon, and the postsynaptic side is the dendrite. As demonstrated in this figure, the presynaptic axon sends signals to other neurons' postsynaptic dendrite. The axon subcellular bouton will send information through a dendrite subcellular spine. Therefore, the more spines a neuron has, the more information it will receive. To conduct my project, we started with injecting biocidin to label the in vivo target cell. Then, the tissue was cut into 50-micron sections to recover the targeted neuron and stained with fluorescent dye to illuminate the target cell. Next, we used the confocal microscope to take 3D image stacks of each tissue section. We uploaded these images into the neuromorph program, which is cited here, but for time's sake, I can't go too far into it. But the main overview of this program was to conduct semi-automated neuron morphology reconstruction pipeline. Over here, we can see the image or the max projection picture we took of the soma of the adult neuron within the visual cortex. As you can recall from the cartoon image, this is what an adult neuron will really look like. This first picture over here is of the neuron's axon. The swelling is attached to the axon, which these arrows highlight, are the neuron's boutons, and this is where the axon will receive or send information to other neurons. This picture over here is of the neuron's dendrite. The arrows are pointing towards the subcellular spines that receive information. This circle over here is highlighting a branch point within the dendrite, where the one singular strand of dendrite was split into two, and this is also known as a bifurcation point. The number of branch points can determine the complexity of a neuron. So to look at the structure of the neuron, we put everything into the neuromorph program, and here we can see that this image is from the neuromorph program. This is a 3D reconstruction of the neuron, and we use this to get the morphology. I was able to go in and connect the axon to the dendrite. So over here, this video shows the neuron morphology that was put into the program and then manually sliced. This structure is in 3D, so we can capture and analyze the complete reconstructed morphology. The red domain, pictured over here, is the neuron's dendrite, and then over here, the blue domain is the neuron's axon. So this image is of the adult neuron dendritic spines, and this is where the cell receives information. The green markers represent the location of the spines along the dendrites that I went through and manually counted using the landmark tool within the Amira platform. Everywhere there is a green marker, the dendrite can receive a signal from another neuron. So all of these over here, these are all places where the dendrite can receive information. To the right, you can see the reconstructed morphology of the adult neuron with all of the labeled spines. For the complete analysis, I went through the multiple stacks of photos to landmark all of these spines. Here, we're now going to look at the complete morphology of the development of a neuron. So this over here is the complete 3D reconstruction of the adult neuron within the visual cortex. These blue lines represent the axon, which put together measures 6.22 centimeters. Yeah, centimeters. So this axon is very large. And the total dendrite length was 6.9 millimeters. And this pink over here represents the apical dendrite, which is the domain of dendrite, which is further away from the cell body. And then over here, we can see the red domain, which is the basal dendrite, which is typically closer to the soma or the cell body. The apical length was 3.15 millimeters, and then the basal length was 3.77 millimeters. So now we can look at the complete reconstruction of a neuron a few days after eye opening, and it is still in its developing stage. The axon was a total length of 2.4 centimeters, or 2.04 centimeters. And again, this is not a spelling mistake, it was actually in centimeters. And then the total dendrite length was 4.75 millimeters, where the apical domain was 1.34 millimeters and the basal was 3.41 millimeters. And I hope you guys are all memorizing these numbers because I will test you later. And then here we can see a reconstruction of a neuron a few days before eye opening. The neuron is still very young, and as you can see, this neuron is significantly smaller with an axon length of 1 centimeter compared with the mature axon length of 6 centimeters. The dendrite total length was 1.95 millimeters, with the apical of 0.94 millimeters and the basal of 1.01 millimeters. So after we got all of these morphological structures, we were able to analyze the neuron. This graph shows that as the neuron develops, the total dendrite path length will elongate. Interestingly, the basal dendrite, seen over here, accounts for almost all of the dendrite growth. This can mean that the apical dendrite matures faster than the basal dendrite. Further, as I mentioned before, branch points can indicate a neuron's complexity. The more branches the neuron has, the less selective, and the less branches the neuron has, the more selective inputs the neuron will sample from. The diminishing number of branch points could be a result of pruning, meaning that as the neuron matures, it seems to become more selective. The total spine count between a developing and mature neuron nearly triples. So as the dendrite elongates and some branches become pruned, the number of spines and density significantly increases. As you can see through this graph on the right, the spine density within the developing neuron nearly doubles. In summary, if one neuron can have 6 centimeters of axon, the neural pill within the brain is very dense. Further, each one of these neurons can receive over 3,000 inputs. This function occurs through the spines highlighted in the image to the right. This is where the dendrite can receive information. Next, as the dendrite matures and their overall path length increases, the dendrite becomes less complex as a result of pruning. We hypothesize that this pruning is a result of the neuron receiving inputs from a more selective group. So, a developing neuron could sample from many different neurons, whereas when it grows, it will become more selective. This would explain the decrease in branch points. The observed dendritic pruning can be evidence of a developmental mechanism whereby neurons in the visual cortex become specific to the inputs that they sample from. The next step in this process would be to recover morphologies from developing neuron in different stages of development, such as right before and right after eye opening. This data would be analyzed with the already recovered adult morphologies to observe correlations between any functional and structural data. After that, this information would allow us to analyze developing neuron morphologies with developing population responses to visual stimuli. I would like to thank Dr. David Fitzpatrick for allowing me to come into his lab and conduct this research, Dr. Mike Guest for being my mentor and helping me along this process, Alex, Ilaria, and Erica for making this program amazing, and then our collaborators, Marcel and Philip, for allowing me to use the Neuromorph and Amira platforms, the Fitzpatrick Lab for welcoming me into their environment and allowing me to learn along with them, and then all of the generous NPFI supporters who made this program absolutely amazing.
And actually, I thought it was a typo, the 6 centimeters. The first time I saw it. Questions for Arielle?
Are these, oh, I didn't realize you had your hand up. Are these axonal lengths in the tree shrew on a much different scale from other small mammals because of their visual cortex importance, or are they similar to lengths of axons in other mammalian species?
So the question was, is the axonal length in tree shrew different from, let's say, rat or other similar animals? And the answer is no, they are actually very similar.
A little different question. As the dendritic spines lengthen, are they reaching toward another neuron, or are they connecting within that neuron?
Yeah, so the question was, as the dendrite elongates, will it connect with the same, its own neuron or another neuron? So as the dendrite elongates and it communicates, it will typically communicate with other cells in the same area.
Any other questions? Yeah. So now I can see a new trend of mentors asking questions.
Could you talk about the variability in the neuron's morphology and how many more neurons would need to be counted for less variability?
Yes, I actually have a slide on that. Surviving.
I was not part of this, I promise.
Although I was able to pull data from these 2 neurons, about 15 neurons will be needed to have more concrete and accurate data in the adult alone to capture 85 to 90% of the anatomical structure. So specifically, about 15 neurons will allow the variability in dendrite morphology to decrease, and the variability within the 15 neurons will be 4%, and within the axon, it'll be like 12%.
Yeah, the question, Mike, or a comment?
Yeah, I know for time's sake, you only focused on the dendrite, but you showed the axon morphology, and you showed this really cool data, like how, you know, the apical dendrite seems to mature first and the basal and this pruning. Do you have any data you could show about the axon? What makes up for like the increase in the axon development?
So the question was to elaborate more on the axon's development, and I actually also have a slide for this. So the axon actually elongates during growth, and surprisingly, the dendrites or the branch points also increase. So whereas in the dendrite, you saw less branch points, in the axon, you will see more branch points.
Okay, thank you so much, Arielle.